Hello everyone and welcome to the second part of the chapter Plant Kingdom. So let's get started with the continued topic Pteridophytes. Pteridophytes. The pteridophytes includes horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues, that is xylem and phloem. You shall study more about these tissues in Chapter 6. The pteridophytes are found in cool, damp, and shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is gametophytic plant body. However, in pteridophytes, the main plant body is sporophyte, which is differentiated into true roots, stems, and leaves. These organs possess well-defined vascular tissues. The leaves in Pteridophyta are small, as in Selaginella, or large, as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophytes. In some cases, sporophytes may form distinct compact structures called strobili or cones. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bears male and female sex organs called antheridia and archegonia respectively. Water is required for transfer of antherozoids, the male gametes released from antheridia, to the mouth of archegonium. Fusion of male gamete with egg present in the archegonium result in the formation of zygote. Zygote thereafter produces multicellular well-defined sporophytes, which is the dominant phase of pteridophytes. In majority of pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro and microspores, are known as heterosporous. The megaspores and the microspores germinate to give rise to female and male gametophytes respectively. The female gametophytes in these plants are retained on the parent sporophyte for variable periods. The development of zygotes into young embryo takes place within the female gametophytes. This event is precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, that contains Psiloptium; Lycopsida, that contains Selaginella and Lycopodium; Sphenopsida, that contains Equisetum; and Pteridopsida, that contains Dapteris, Pteris, and Adiantum. Gymnosperms. The gymnosperms, that is naked seeds, are plants in which the ovules are not enclosed by any ovary wall and remains exposed both before and after fertilization. The seeds that develop post-fertilization are not covered, that is, are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. One of the gymnosperms, the giant redwood tree, sequoia, is one of the tallest tree species. The roots are generally taproots. Roots in some genera have fungal association in the form of mycorrhiza, that is in Pinus, while in some others, that is in Cycas, small specialized roots called colloid roots are associated with the antofixing cyanobacteria. The stems are unbranched or branched. The leaves may be simple or compound. In Cycas, the pinnate leaves persist for a few years. Leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form legs or compact strobili or cones. The strobili bearing microsporophylls and microsporangia are called microsporogenate or male strobili. The microspores develop into a male gametophytic generation, which is highly reduced and is confined to only limited number of cells. This reduced gametophyte is called pollen grain. The development of pollen grain takes place within the microsporangia. The cones bearing microsporophylls with ovules or microsporangia are called microsporogenate or female strobili. The male or female cones or strobili may be borne on the same tree, that is in Pinus. However, in Ficus, male cones and megasporophytes are borne on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and the composite structure is called the ovule. The ovules are borne on megasporophytes which may be clustered to form the female cones. The megaspore mother cells divide meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within the megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms, the male and female gametophytes do not have an independent free-living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of ovules borne on megasporophytes. The pollen tube carrying the male gametes grows towards archegonia in the ovules and discharge their contents near to the mouth of archegonia. Following fertilization, zygote develops into an embryo and the ovules into the seeds. These seeds are not covered. Angiosperms. Unlike the gymnosperms where the ovules are naked, in angiosperms or flowering plants, the pollen grains and the ovules are developed in specialized structures called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in a wide range of habitats. They range in size from smallest wolffia to the tall trees of eucalyptus that is over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into two classes, the dicotyledons and the monocotyledons. The dicotyledons are characterized by seeds having two cotyledons, reticulate venetions in leaves, and the tetramerous or pentamerous flowers, that is, having 4 or 5 members in each floral walls. The monocotyledons, on the other hand, are characterized by single cotyledonous seeds, parallel venation in the leaves, and trimerous flowers having 3 members in each floral wall. The male sex organ in flower is stamen. Each stamen consists of a slender filament with an anther at the tip. Within the anthers, the pollen mother cell divides by meiosis to produce microspores which matures into pollen grains. The female sex organ in flower is pistil. The pistil consists of a swollen ovary at its base, a long slender style, and stigma. Inside ovary, ovules are present. Generally, each ovule has a megaspore mother cell that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 divide to form embryo sac. Each embryo sac has 3-celled egg apparatus, 1 egg cell and 2 synergids, 3 antipodal cells and 2 polar nuclei. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain, after dispersal from anthers, are carried by wind or other agencies to stigma of a pistil. This is termed as pollination. The pollen grains germinates on stigma and the resulting pollen tubes grow through the tissues of stigma and style and reach the ovule. The pollen tubes enters the embryo sac where two male gametes are discharged. One of the male gamete fuses with the egg cell, that is syngamy, to form zygote, and the other male gamete fuses with the diploid secondary nucleus to produce triploid primary endosperm nucleus, that is PEN. Because of the occurrence of two fusions, that is syngamy and triple fusion, this event is termed as double fertilization, which is an event unique to angiosperms. The zygote develops into an embryo, that is with one or two cotyledons, and the PEN develops into endosperm, which provides nourishment to the developing embryo. The synergids and the antipodal degenerates after fertilization. During these events, the ovule develops into seeds and the ovaries develops into fruit. The life cycle of an angiosperm is shown in figure 3.6. Plant life cycles and alteration of generations. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of an sexually reproducing plant, there is an alteration of generations between gametes producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in following patterns. First, sporophytic generation is represented only by one cell zygote. There are no free-living sporophytes. Meiosis in the zygotes result in formation of haploid spores. The haploid spores divide mitotically and form gametophyte. The dominant photosynthetic phase in such plants is free-living gametophyte. This kind of life cycle is termed as haplontic. Many algae such as volvox, Spirogyra, and some species of Chlamydomonas represent this pattern. Second, on the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by a single to few-celled haploid gametophyte. This kind of life cycle is termed as diplontic. An alga, Fucus, represents this pattern. In addition, all seed-bearing plants, that is, gymnosperms and angiosperms, follow this pattern with some variations wherein the gametophytic phase is few to multicelled. Third, bryophytes and pteridophytes interestingly exhibits an intermediate condition, that is haplodiplontic. Both phases are multicellular. However, they differ in their dominant phases. A dominant independent photosynthetic thalloid or erect phase is represented by haploid gametophyte, and it alternates with the short-lived multicellular sporophyte, totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represents this pattern. The diploid sporophyte is represented by a dominant, independent, photosynthetic, vascular plant body. It alternates with multicellular, sporophytic or autotrophic, independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycles. All pteridophytes exhibits this pattern. Interestingly, while most alga genera are haplontic, some of them such as Ectocarpus, Polysymphonia, Kelps are haplodiplontic. Fucus, an alga, is diplontic. So, we are done with the third chapter of biology, Plant Kingdom. I hope it helped you to listen and learn. And don't forget to like, subscribe and comment. And if you find this useful, share it with your friends. Bye bye.